to release catecholamines such as norepinephrine and epinephrine with salivary alpha amylase, a digestive enzyme found in oral cavity serving as marker for SNS activity. Elevated levels of both alpha amylase and cortisol have been indicated following various stressors. Reproduction is regulated by the secretion of sex steroids, testosterone, estrogen, and progesterone through the HPGA. As each of these functions require considerable physiological resources, it is not surprising that the neuroendocrine systems that regulate them are interrelated, allowing for mutual modulation according to specific environmental and internal conditions. To the best of our knowledge, only few studies address the HPA-HPG axis crosstalk regarding cognition and directly measured hormonal levels. For example, Juster and colleagues revealed sex-specific association between sex hormones and cortisol reactivity. However, they did not study the SNS-HPG axis interconnectivity. Therefore, the first aim of our study was to investigate the role of sex hormones in modulating stress reactivity. Previous studies examined the impact of stress exposure on cognitive performance. In particular, they have focused on memory and on visuospatial abilities. The literature on the effects of acute stress and on the association between cortisol reactivity and declarative memory showed inconsistent findings. The literature on the effects of acute stress and cortisol reactivity on visuospatial performance show inconsistent findings as well. These discrepancies may be explained by methodological differences, such as the memory and visuospatial testing procedure used, or the time of testing, or differences in stress procedures. Some used parachute jumping, others used physical exercise or physical challenges, such as the cold pressure task, or psychological challenges, such as mental task or psychosocial task, such as the Trier Social Stress Test, the TSST. The present study aimed at systematically examining the effects of psychosocial stress on memory and on visuospatial performance using the TSST. Aside from the methodological differences, the discrepancies may be explained also through involvement of sex hormones. Given that the neurobiological mechanisms underlying reproduction and stress response are interlinked, sex hormones may modulate the effects of stress in general and on visuospatial and memory capabilities in particular. Previous studies examining the relation between hormone level and individual differences in cognitive abilities produce inconsistent findings. For example, some studies showed a positive relationship between testosterone and spatial abilities, while others failed to produce significant correlations or showed negative or even curvilinear relationships. Only few studies addressed the role of estrogen and progesterone in individual differences in cognitive abilities, and they also produced mixed findings. Some have documented positive relationship between estrogen and progesterone and verbal memory, while others fail to support this pattern of findings. Therefore, the third aim of the present study was to examine the interaction between stress systems, SNS and HPAA, and baseline reproductive hormones in moderating the effects of psychosocial stress on declarative memory and on visuospatial abilities. The study sample included 112 young participants. 39 of them were men. Of the women, 37 were taking oral contraceptive, the OC group. The remaining 36 women were not using oral contraceptives and were in the mid-luteal phase, day 21, of their menstrual cycle at the time of the study, the LP group. The cognitive battery task included 2 tasks. Visuospatial performance was measured by the mental rotation task. In the MRT, there were 5 3D stimuli presented on the screen. One was the target stimulus, 2 of the non-target stimuli were identical to the target, and another 2 were almost identical. The participants were instructed to decide which of the non-target stimuli were identical to the target stimulus by mentally rotating them. Declarative memory was measured by the Ray Auditory Verbal Learning Test. The result is composed of 7 trials. On each of the first 5 consecutive trials, an experimenter read a list of 15 common nouns to the participants. Each reading was followed by a free recall task in which participants were asked to repeat as many of those words as possible. In trial 6, an interference list was presented, and in trial 7, participants were asked to recall the words from the first list. The biological markers were the reproductive hormones, estradiol, progesterone, and testosterone, and stress biomarkers, cortisol and alpha amylase. Participants were randomly assigned to one of 2 groups, stress, using the TSST procedure, and non-stress. The experimental session took place in the laboratory of the psychology department between 8 to 8:30 at morning, a time period during which testosterone levels were at a higher peak. All participants were tested at least 1 hour after awakening to avoid the possibility that the cortisol increase would be due awakening response. Participants from each group, men, OC, and LP, were randomly assigned to one of 2 experimental groups, stress or control. The experimental session was comprised of 3 consecutive stages: the result and the MRT, the TSST or the control condition, and the second completion of the result and the MRT. The participant provided saliva samples at 4 assessment points: T1, baseline, T2, immediately following the TSST or control condition, and T3 and T4. The T1 sample evaluated levels of testosterone, estradiol, and progesterone, as well as baseline levels of cortisol and alpha amylase. The remaining samples evaluated levels of reactive cortisol and alpha amylase. For cortisol levels, the following analysis was conducted separately for responders and non-responders. For responders, a 2-way repeated measure analysis of variance demonstrated no significant main effect for group. That is, there was no significant difference in cortisol secretion between men, OC, and LP groups. A significant main effect was found for time, with post-hoc analysis revealing that cortisol levels at T3 were higher than other cortisol measurements. However, in reanalysis controlling for sex hormones, the main effect for time disappeared. For alpha amylase, a 2-way repeated measure analysis for variance demonstrated no significant main effect for group. A significant main effect was found for time, with post-hoc analysis revealing that alpha amylase levels at T2 were higher than the other measurements. However, in reanalyzing controlling for sex hormones, the main effect for time disappeared. Examining the reactivity patterns of the 2 biomarkers for stress show unique reactivity patterns for each biomarker. While a peak was observed for cortisol on T3, as you can see over here, followed by a decline in cortisol level, for alpha amylase, an immediate elevation was apparent and conservation of elevated levels was demonstrated, perhaps given the continuity of stress induced by the cognitive task. The differences between the 2 main stress systems in the time points following stress agree with previous findings. The SNS releases catecholamines immediately at the onset of a stressor, whereas the slower active HPA axis releases glucocorticoids 21 to 45 minutes following the onset of a stressor. Regarding the influence of stress exposure on memory performance, a significant main effect was found for memory trial, and with the post-hoc analysis revealing a positive learning curve occurring from trial 1 to trial 5. A significant main effect for stress was found as well, such that the group exposed to the TSST demonstrated poor recovery after interference as compared to the control group. Regarding the influence of stress exposure on mental rotation performance, there was a significant stress by time interaction, and decomposing this interaction revealed that in the control group no significant difference in performance was found before and after exposure to the non-stress condition. However, for the stress group, performance on the mental rotation improved significantly after the TSST. Regarding the interaction between reproductive hormones and stress system in the effects of stress on declarative memory, in reanalysis controlling for sex hormone, the main effect of stress disappeared. For further analysis, cortisol and alpha amylase were calculated as the increase from baseline to peak. T3 for delta C and T2 for delta alpha amylase. The analysis revealed that there was a significant two-way testosterone by delta C interaction in predicting the difference in memory performance before and after stress exposure, and there was a significant two-way progesterone by alpha amylase interaction in predicting the difference in memory performance before and after stress. Regarding the interaction between reproductive hormones and stress systems and the effect of stress on mental rotation, in reanalysis controlling for sex hormones, the main effect for stress disappeared. Further analysis revealed that estrogen was correlated with the difference in mental rotation performance before and after stress exposure. Furthermore, among men, delta C was positively correlated with the difference in mental rotation performance before and after stress. In sum, reproductive hormones influence the activation of the two major stress systems, the HPA-A and the SNS. These results emphasize the need to control for sex hormones fluctuation when examining stress reactivity. Furthermore, psychosocial stress differentially influences cognitive performance. It enhances mental rotation performance where it disrupts aspects of declarative memory. Future studies should examine the influence of stress exposure on other cognitive domains and other stressors. Last, these moderating effects appear to depend on the activity of reproductive hormones. This novel finding strengthened the importance of HPA-HPG interaction on behavior and are come among the first demonstration on the role of SNS-HPG interaction on cognitive function. Thank you very much for your attention.